Good morning, science class. What we're going to talk about today is diffusion. What is diffusion? Well, we know this from the first part is diffusion is going to be particles moving from an area of high concentration to an area of low concentration across the semi-permeable membrane. So what I'm going to do today is we're going to show how can we actually model that in a lab. We know this is occurring between cells, but how does it actually work? So here we go. The first part of the lab that we're going to talk about is we're going to be using a thing called iodine. Iodine it looks just like this. You can see it's kind of that reddish yellow tint, and that's a pretty high concentration of iodine I have in there. The other thing we're going to use today is we're going to use just your run-of-the-mill good old cornstarch that you get in any house probably across America. And I'm going to break it into two pieces today. I'm going to have, and the first part is I'm going to talk about how is iodine an indicator. It's an indicator for starch. So what does that actually mean? That means this. So I'm going to take a beaker with just water. What I'm going to do is I'm going to put starch in it. And of course, I'm going to sit here and I'm going to mix it around. It looks kind of almost like milk. And I'm just going to put and make sure there's enough starch. I want a lot in there. And this is just to show how does iodine indicate in the presence of starch. All right, so what I'm going to do is I'm going to mix this up just like this. Now, what I'm going to do with the iodine is I'm going to put drops in there and we're going to see what happens. How do I know that there's starch present in this? What happens to the color? And as you look at that, you can see it starts to change colors from white to purple. And what is that telling me? That's telling me that the iodine is saying, hey, there's starch in here. That's what an indicator is. So iodine is an indicator for starch. It'll change color in the presence of it. All right. Now, the second part of this is how are we going to show diffusion in the lab? Well, here we go. I'm going to take your average run-of-the-mill regular just sandwich bag. People probably all have these at home. I'm going to put in cornstarch in here. Oh, I missed. Let's try that again. Cornstarch in the bag. Oh, I'm making a mess. That's all right. Messes are good. Messes mean you've done something, right? So here's my bag. I got, you can see, just some cornstarch in there. What I'm going to do then is I'm just going to pour some water in the bag. And now I have a bag with just milky white, you can see cornstarch. So what am I going to do with that? I'm going to mix it up, have it all mixed up, and I'm going to tie the bag. And all I'm going to do is I'm going to tie it in a knot. Simple as that. And once I have it tied, there it is. There is going to represent the cell. We're going to say this is the cell. The bag is our selectively permeable membrane or semi-permeable, depends the way on however it means the same thing. So it means it's going to allow certain things in and certain things out. So we're going to see what happens to this. Okay, so what am I going to do with that? Well, the next thing I'm going to do is I'm just going to take my water and I'm going to put iodine in it. And you can see how that changes the color iodine. Now, here's my iodine, here's my starch. How do I know if a particle moves? If a particle moves, when I put this in here and let it soak, if a particle moves, what will happen? If the iodine moves, what's going to happen to the color of this bag, inside the bag? It's going to turn purple. And if the cornstarch moves out, the iodine will turn purple. So we'll be able to tell after 15 minutes which particle moves. Okay? And all I'm going to do is I'm going to put this in here for 15 minutes. Now, things you want to think about here, and that's when you're working through that sheet, is for the next 15 minutes, you're going to be working on your own. Then you're going to come back in class, and I'm going to show you the final result, and we can discuss what happened and why. And to me, You don't think of particles moving. You don't think of them having this energy to move from a high concentration to low. We just assume it happens, but we're going to look and see it today. So when you finish this video and you have your sheet all done, come back into the classroom and we'll see what happens next. All right, cool.